Alright, in this video we are going to be talking about lipids. Alright, lipids are a type of compounds. These are organic compounds of biological origin that are insoluble in water and soluble in nonpolar solvents. So if you think about what we've been doing this entire semester, we've really been organizing the material we've covered based on functional groups. We've been learning about different functional groups, nomenclature, concepts, reactions, and mechanisms. But for lipids, we're really going to be focusing on organizing these compounds not based upon any particular functional group, not based upon function, but really focused on a physical property. So lipids are classified as lipids based on a physical property, and that physical property is really solubility. So these compounds are organized and classified based on solubility. So what that means is these compounds are going to have a variety of different functional groups, a variety of different functions, but what they have in common is that they are insoluble in water and soluble in nonpolar solvents. These properties are usually due to large nonpolar regions. So we're going to see later an example called a phospholipid. If you see this compound, it has a really large region that is nonpolar, and that really drives the solubility. So a couple of terms we need to know and understand. The first is hydrophobic. Compounds that are hydrophobic are water-hating. They're insoluble in water. And lipophilic. Lipophilic are compounds that are fat-loving or are soluble in nonpolar solvents. So our lipids are in fact hydrophobic and lipophilic. The opposite of that, of course, is hydrophilic and lipophobic, which is not the properties that lipids have. All right. Lipids are found in fatty tissues and other non-biological structures such as membranes, and there's generally 8 different classifications. The first classification is a fatty acid. The second is a wax. The third classification is a triacylglycerol. Fourth are phospholipids. 5 prostaglandins. 6 steroids. And 7 are vitamins. The 8th classification are terpenes. We are not going to be discussing terpenes in this lecture. So we're really going to focus on 7 different types of lipids. So what we'll do for the rest of this video is really walk through each of these classifications. And with each of these compounds, we want to have a general idea of the structure and, in some cases, understand the properties of those. So our focus here is really understanding structure, and then occasionally we'll talk about some properties or some function of these compounds. Alright, so let's talk about the first class here. The first class are fatty acids, and this is pretty simple. These are unbranched carboxylic acids. So you have to have the carboxylic acid functional group, which you see in these compounds here. And then the other key to these compounds is they have to have 12 to 20 carbons, and they have to have an even number of carbons. So fatty acids are carboxylic acids that are typically contain 12 to 20 carbons, but it must be an even number. That even number arises because how they're biosynthesized, 2 carbons are always added at a time. The most important fatty acids have 18 carbons. And again, I just want to point out that the carbon of the carboxylic acid counts. So if we count our carbons for lauric acid, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12, the carbon of the carboxylic acid counts as one of the carbons. Alright? So you can have saturated fatty acids. Saturated fatty acids do not have any carbon-carbon double bonds. And here we see a bunch of examples, 12 carbons, 14, 16, 18, or 20. You do not need to know these specific names, you just need to understand the general class. Fatty acids, carboxylic acid, even number of carbons. So you can also have unsaturated fatty acids. Unsaturated fatty acids contain carbon chains that have one or more carbon-carbon double bond. It turns out the cis carbon-carbon double bond is more common than the trans. So here are 4 more examples of fatty acids, and these happen to be unsaturated fatty acids because they contain alkenes in the long carbon chain. Again, carboxylic acid, functional group, even number of carbons. Unsaturated means they contain carbon-carbon double bonds. All right, polyunsaturated fatty acids obviously have 2 or more carbon-carbon double bonds. All right, next let's talk about the next class here. Those are compounds classified as waxes. A wax is simply an ester derived from fatty acid and a long alcohol. So an ester simply has, a wax is simply an ester that has 2 long carbon chains. Again, the synthesis of these is done with a long chain alcohol and a fatty acid. So again, the fatty acid has to have an even number of carbons. And typically, the long chain alcohols also have an even number of carbons. All right? So, very simple. Ester functional group, even number of carbons. Again, don't forget the carbon of the carbonyl counts for the even number of carbons when you're drawing an ester. The main biological function of waxes is as a water barrier, right? These are the type of compounds that are used in car waxes. They're found in feathers to prevent wetting, also in leaves to prevent water evaporation. All right, the third type of compound is called a triacylglycerol or a triglyceride. Triacylglycerol or triglyceride. These are fatty acid triesters made from glycerol. All right, here's the structure of glycerol. It contains 3 carbons and 3 hydroxy groups. Glycerol combines or reacts with fatty acids. Again, these compounds have to have an even number of carbons, and those are produced in the body to form a triacylglycerol. So let's just draw the structure of these so we're clear. There's the glycerol derivative. Instead of alcohols, we have ester functional groups. O, C double bond O, R, O, C double bond O, R, O, C double bond O, R. And these compounds at the end are derived from the fatty acids, which means they have to have an even number of carbons. Again, typically, fatty acids, the most common one has 18 carbons. So you'll see glycerol reacting with fatty acids that have 18 total carbons. And there's simply 3 of those. These are the most abundant natural lipids. Okay, they're the most abundant natural lipids. The main biological function is they are used for energy storage. All right. So those are 2 things we should definitely highlight and remember. Triacylglycerols are the most abundant natural lipids, and the main biological function is used for energy storage. Interesting, the hydrolysis of this reaction of animal fats yields soap. And what I want to point out is triglycerides are called fats if they are solid at room temperature. These are oils if they're liquids at room temperature. So if you take a fat or an oil and hydrolyze this, you yield a soap molecule. And let's remember this hydrolysis reaction where we cleave an ester under basic conditions is called saponification. All right, saponification will yield one equivalent of glycerol and then get you back to your fatty acid. All right, so essentially fatty acids can be used as soaps. So let's talk a little bit about how soaps work. All right, soaps work because they have a long nonpolar hydrocarbon chain, and that nonpolar hydrocarbon chain will be attracted to grease or an oil. All right, micelles, another key word we should know, micelles result, these are spherical aggregates. They're basically giant spheres that are hydrophilic on the outside because of the carboxylic acid derivatives, and they are hydrophobic on the inside. All right, so micelles formed from soap are hydrophilic on the outside, hydrophobic on the inside. And essentially what happens is you have a lump of oil or dirt, your soaps are going to adsorb into it. The long carbon chains will go into the nonpolar portion of that oil. They will then form a micelle which solubilizes your stain, right? So the hydrophobic tail will be attracted to the hydrophobic oil or dirt. The hydrophilic head will be exposed to water, right? That's normally the solvent used when you're washing something. And you form a micelle, which then lifts the stain off a shirt or whatever you're doing. It really carries that grease oil away when that wash water is removed. And that's how soaps work. If you've ever seen the movie Fight Club, Tyler Durden, right, his profession is making soap from fat. And that is an accurate chemical reaction that can take place. If you take fat and hydrolyze it, right, you can produce different soap molecules. All right, one thing to consider with soaps is a lot of water contains a lot of inorganic salts such as calcium, magnesium, and iron. These salts will bind with the carboxylate of the fatty acids, which gives them poor solubility. So synthetic soaps, soaps that we actually use sulfonates, and these sulfonates are less likely to bind with calcium, magnesium, and iron. So this compound here, sodium 4-dodecylbenzene sulfonate, is a typical synthetic soap used in detergents, washing hands, things like that. All right, let's talk about our next class of molecules. These are called phospholipids. Phospholipids are again glycerol triesters. So here is our glycerol molecule. All right, let's highlight. There's your glycerol molecule. But now these are attached to two fatty acids. All right, that's a C double bond O. But then one phosphate linkage. So phospholipids are glycerol triesters formed from two fatty acids. There's the two fatty acids and one phosphate group. All right? The fatty acids are typically contain C16, C18 carbons. These are the second most abundant lipid. All right? What we see with these compounds is that the long tails derived from the fatty acids are hydrophobic. These hydrophobic tails really want to avoid water, but they will associate with each other, resulting in what we have phospholipid bilayers. all right? And the main biological function of phospholipids is they are used as cell membranes. So let's investigate what a cell membrane looks in a little more detail. In this figure, we have a simplified view of a cell membrane. So these are made up from our phospholipid compounds. What you see is you have the hydrophobic heads. These are formed from the phosphorus groups. So these are all compounds that are polar, and they will form on the outside of the cell in an aqueous environment. And what we know on the inside of a cell is also an aqueous environment. So we see they form this lipid bilayer. All right. What that means is we have one lipid at the top, long chain, another long chain, your phospholipid at the bottom. So the key here is that the hydrophobic regions are now interacting with each other to form this membrane. All right. So cells are really protected because it's hard to cross this membrane, right? A compound that is nonpolar is going to tend to get stuck in the hydrophobic tail portion within the main membrane. Something that's hydrophilic will be on the exterior of the cell or the interior of the cell. Okay, so the fifth lipid we need to talk about are called prostaglandins. Prostaglandins. These are molecules that have the prostanoic acid skeleton, and that is really the structure you need to know, which is right here. So you don't need to know any specific examples, you just need to know the basic skeleton. And in this basic skeleton, you have a cyclopentane ring. There are 7 carbons attached to carbon 1. Those 7 carbons end in a carboxylic acid. And then at carbon 2 of the cyclopentane derivative, there are 8 carbons. So for prostaglandins, you just need to know the prostanoic acid skeleton structure. All right. The other thing you should be familiar with is that these molecules have typical half-lives that are 5 minutes or less. So what that means is the synthesis of these compounds occurs at the point of need. For example, a wound site. So here's one example, prostaglandin F2 alpha or PGF2 alpha. And here you can see you have the prostanoic acid skeleton, a cyclopentane ring, 7 carbons ending in a carboxylic acid, and then an 8 carbon chain. And this just shows you that there is derivatization of this skeleton structure. Typically what you find is alkenes and alcohols that have stereochemistry on it. So this is just showing you the biosynthesis of these compounds. They're really made from arachidonic acid, and using different enzymes in multiple steps, different prostaglandins can be formed. So here's an example here, another example there, and you can see all of these compounds have that same skeleton structure. You can have different alcohols, carbonyls, different alkenes in these molecules. The biological function of these is widely variable. They can be used as platelet aggregation inhibitors. They can regulate blood pressure, right, or stimulate muscles as well. So again, similar structures here, but a wide variety of functions. And again, our focus here is understanding the skeletal structure. Okay, the sixth lipid we want to talk about are steroids. These are molecules that have the tetracyclic carbon skeleton structure that's shown. And again, I just want you to be familiar with the steroid ring skeleton. There are 4 rings in this, A, B, C, D, 3 cyclohexane rings, and 1 cyclopentane ring. So you just need to be familiar with the steroid ring skeleton structure. A great example of a steroid is cholesterol, right? These molecules are flat and rigid. If you typically look at the stereochemistry, here's the first ring, there's B ring, C ring, and D ring. Because of the stereochemistry around these molecules tend to be pretty flat and also fairly rigid. Again, different steroids are synthesized via biosynthetic steps. The formation of cholesterol takes more than 60 steps, right? So these are pretty complex molecules and take a lot of steps to be synthesized in our bodies. Cholesterol is a biological precursor for all of the other steroids. Here are a few examples. They can be used as sex hormones, such as testosterone and estrone. These can be other hormones involved in metabolic processes, like cortisone and aldosterone. And then you can have steroids that really aid in digestion. An example of that is cholic acid. So when you hear the term steroid, you probably think about anabolic steroids, right? And that is just one class of steroids. All right, steroids naturally occur in our bodies. They're used, as we see, as hormones and digestions. But these are compounds that have been historically sort of abused to aid, their really performance-enhancing drugs. All right, but cholesterol is a steroid found in our body, produced in our body. It's a natural steroid. So we need to understand this definition from a chemistry standpoint, that not all steroids are anabolic steroids. They're not all illegal. In fact, they're crucial hormones that our body uses. So the last example we're going to be talking about are lipophilic vitamins. So this is a very generic class. This is really sort of a catch-all category. A vitamin is an organic compound that's not a fat, not a protein, not a carbohydrate, that's required for normal growth and maintenance of animals. So it's basically an organic compound that's not any of these other classifications that our bodies need. They have a very, very broad range of origins, structures, and functions. All right. So for this category, it's really sort of a catch-all. There's a lot of different structures of vitamins. They have a lot of different biological functions. So let's just look at a few of them. One example is vitamin E. This is a lipophilic antioxidant. It prevents oxidative cellular damage from radicals. There are D vitamins. These regulate calcium and phosphorus metabolism. All right, and you can see that sort of looks like a steroid, but because it doesn't have a B ring, it's not. And another example is vitamin A, or retinol. This is a vitamin that's essential to vision. It's incorporated in the light harvesting portion of rhodopsin, which is a vision protein. So you can see from these structures that vitamins really have a lot of different structures. Again, it's this catch-all category of organic compounds that bodies need, but there's not really sort of one class of structure for these. All right. So that really concludes our lecture. We've talked about 7 main lipids. Generally speaking, you should be able to understand what a typical molecule in each classification looks like, or the skeletal structure, and then in cases where I highlighted it, understand sort of the function.